Responses to the End of Cycle Examinations 2025 Term 3. Item 1. In a Southeast Asian coastal village, illegal cyanide fishing is practiced. Fishermen capture live fish for aquariums. This has led to a significant die-off of non-target coral reef species. A marine biologist, Dr. Lim, collects tissue samples from affected damselfish. Cyanide is a potent inhibitor of cytochrome C oxidase. Analyze the table. Task. Part A. Explain, in simple steps, how cyanide inhibition of cytochrome C oxidase leads to the changes shown in table. Part B. Using the information provided, propose strategies to solve the challenges. Cyanide blocks cytochrome C oxidase, the enzyme that allows electrons to move to oxygen at the end of the electron transport chain. When the enzyme is blocked, aerobic respiration stops, so mitochondria cannot use oxygen to make ATP. As a result, ATP levels fall sharply from 4.8 in normal fish to 0.8 in exposed fish. With little ATP, energy-dependent pumps on the gill cell membrane stop working. These pumps control ions and water movement in and out of cells. The failure of these pumps causes cells to swell, burst, and lose membrane integrity, shown by the rise in damaged cells from 5% to 60%. Because mitochondria do not use oxygen, oxygen uptake rate falls from 9.5 to 1.2. Altogether, cyanide poisoning stops energy production, damages gill cells, and prevents the fish from breathing properly. Stops cyanide use immediately, prevents further enzyme blockage and ATP loss. When cytochrome C oxidase activity fell from 100% to 3%, ATP dropped from 4.8 to 0.8. If fishing stops, enzyme activity in surviving fish can return toward normal. Rescue and stabilize affected fish in oxygen-rich, clean water. High oxygen levels allow remaining mitochondria to resume limited respiration. Oxygen uptake in poisoned fish is only 1.2 with 9.5. Increasing available oxygen helps raise this rate and improve ATP levels. Repair and monitor reef recovery using biochemical tests. Measuring enzyme activity, ATP, and cell damage over time shows if recovery is occurring. These same indicators in the table, enzyme, ATP, cell damage, clearly show cyanide injury, so they can also show healing when values move back toward normal. Educate fishermen using scientific demonstrations. Show how cyanide stops ATP production and damages cells using posters or short videos in the local language. Explain that reefs with healthy fish provide long-term income, while cyanide kills the source of future catches. Item 2. In a school greenhouse, maize, C4 plant, developed yellow patches and stunted growth under prolonged artificial light. Temperature fluctuated widely, and carbon dioxide concentration was low. Microscopy revealed partial loss of Cran's anatomy and reduced PP carboxylase activity. Analyze the data table. IT. Task. Part A. Roman numeral 1. Analyze how light duration, temperature fluctuation, and carbon dioxide concentration collectively affect C4 enzyme activities and chloroplast structure. Roman numeral 2. Explain how changes in these variables influence macromolecule synthesis, starch, proteins, lipids, and stomatal regulation. Too much light, 20 hours per day, causes light stress. Chlorophyll absorbs more light than it can use, damaging thylakoids and reducing chlorophyll content from 2.8 to 1.2. Large temperature variations, between 38 degrees Celsius per day with 15 degrees Celsius at night, denature enzymes at high temperatures and slow reactions at low temperatures. This lowers both PP carboxylase and rubisco activities. Low carbon dioxide limits the substrate for carbon fixation. PEP carboxylase captures less carbon dioxide, and rubisco receives less concentrated carbon dioxide, so photosynthesis drops. Therefore, prolonged light, unstable temperature, and low carbon dioxide damage chloroplasts and Cran's anatomy, exposing rubisco to oxygen concentrations leading to photorespiration, greatly reducing photosynthetic enzyme efficiency. Less carbon dioxide fixation means less sugar for making starch, proteins, and lipids. Low energy and carbon supply slow growth and cause yellow leaves. Stress causes stomata to close to prevent water loss, reducing guard cell aperture from 4.2 to 1.2 and further limiting carbon dioxide entry. Altogether, photosynthesis, biosynthesis, and gas exchange collapse, leading to stunted chlorotic plants. Correct light duration. Use timers for 12 hours of moderate light and 12 hours of darkness to allow chloroplast repair. Stabilize temperature. Maintain 25 to 28 degrees Celsius using shade cloth for heat and insulation at night to prevent enzyme damage. Raise carbon dioxide to normal. Use controlled ventilation or safe carbon dioxide enrichment to support PEP and rubisco activity. Relieve plant stress. Water evenly and apply balanced fertilizer to rebuild chlorophyll and proteins. Monitor recovery. Check leaf greenness and growth. Restored chlorophyll and enzyme activity show photosynthesis improving. Item 3. In a busy Kampala hospital, patients in the COVID-19 recovery ward were monitored for oxygen levels and immune response. 3 patients, Rita, Sam, and Fiona, were studied. Fiona developed slight facial swelling after the second vaccine dose, but recovered quickly. Sam developed mild dehydration symptoms and an elevated heart rate. Analyze the table. Task, Part A. Explain how immunity and homeostasis account for the differences in recovery and physiological data among the 3 patients. Part B. Describe how antibodies and ADH helped Rita and Fiona maintain stability compared to Sam. Part C. Propose safe management strategies to enhance recovery in similar patients. Rita and Fiona are fully vaccinated and show high antibody titers. These antibodies neutralize virus particles quickly. Sam's titer is very low, so his immune system is slower to control the infection. Sam has the highest white blood cell count and the highest temperature. This pattern shows active infection and inflammation. Rita and Fiona have lower white blood cell counts and near normal temperatures, consistent with less ongoing infection. Oxygenation and organ function. Rita and Fiona maintain higher blood oxygen because their lungs are less affected. Sam's oxygen is lower, reflecting more respiratory stress. Homeostatic responses. Sam's ADH is elevated, which indicates dehydration or a strong fluid-conserving response. Rita and Fiona have normal ADH. Elevated ADH helps keep blood volume up by causing water reabsorption, but also signals that Sam is physiologically stressed. Antibodies. They bind and neutralize SARS-CoV-2, stopping virus cells from infecting more tissue. Rita's very high titer, 1,000, and Fiona's moderate high, 620, reduce viral load early, so less lung damage and less inflammation, higher oxygen, and lower fever. Sam's low titer, 40, gives poor early protection, allowing more viral replication and stronger symptoms. ADH, water balance. Normal ADH in Rita and Fiona means their fluid balance is stable, so blood pressure and circulation are steady and oxygen delivery is preserved. Sam's high ADH shows his body is trying to conserve water because of dehydration or stress. This is a compensatory response, but also a sign of instability that requires correction. Full vaccination stimulates adaptive immunity to form memory B and T cells, which produce antibodies quickly upon reinfection. This prevents severe symptoms and helps the body clear the virus faster, as seen in Rita and Fiona. Adequate hydration restores water balance, maintains blood pressure, and allows efficient transport of oxygen and nutrients. It also lowers ADH secretion to normal levels, helping stabilize body fluids and temperature. Supplemental oxygen therapy increases oxygen availability for cellular respiration, reducing fatigue and promoting faster healing of body tissues, especially in patients like Sam with low oxygen saturation. Balanced nutrition provides proteins for making antibodies and repairing tissues, and vitamins, like C and D, to strengthen immune cells. This speeds up recovery and maintains energy balance. Adequate rest and light physical activity. Rest reduces metabolic stress and allows the immune system to focus on fighting infection, while mild exercise improves blood circulation and oxygen delivery to cells, supporting full recovery. Item 4. At Kasubi High School, the biology teacher organized a health awareness week. Students learned about the importance of vaccination, good nutrition, personal hygiene, and physical fitness. During one session, a demonstration was done to compare immune responses between vaccinated and unvaccinated students after exposure to a harmless simulated pathogen. After the activity, teachers encouraged students to maintain healthy habits such as eating well, keeping dormitories clean, getting enough rest, and taking part in morning exercises to boost their general body immunity. Analyze the data table. Task. Part A. Explain the difference between innate and adaptive immunity shown in this demonstration. Part B. Describe how vaccination prepares the body to respond quickly during infection. Part C. Suggest school-level health measures that support immunity among students. In the demonstration, the unvaccinated students relied first on innate defenses, so their response was slower. Reaction time was 5 days, and they developed symptoms like higher temperature, 38.2 degrees Celsius. The vaccinated students showed a rapid adaptive response time of 1 day, presence of memory B cells/white blood cells that rapidly multiply to form plasma cells that form high-concentration antibodies in the body to fight or neutralize the antigen before any damage to the body tissues, hence no symptoms. A vaccine exposes the immune system to a safe form or part of the pathogen, a weakened or killed antigen, protein, or genetic material. This triggers a primary adaptive response: activation of B cells and T cells, production of antibodies, and formation of memory B and T cells. On real exposure, those memory cells initiate a secondary response that is faster and stronger. Memory B cells rapidly produce large amounts of specific antibodies, which neutralize the virus and help phagocytes clear it. This quick, high-level response prevents symptoms, which explains vaccinated students' normal temperature and absence of disease. School-level health measures that support immunity. Regular vaccination of students stimulates adaptive immunity and forms memory cells that protect against future infections. A balanced diet in school meals provides essential nutrients like proteins and vitamins needed for making antibodies and immune cells. Maintaining a clean school environment reduces exposure to disease-causing microorganisms, lowering infection risk. Encouraging adequate rest and sleep allows the body to recover and maintain effective immune responses. Engaging in regular physical exercise improves blood circulation, helping immune cells reach all parts of the body efficiently.